All right. I don't know if I'm pronouncing that correctly, but it looks like you might be our audience today. So we'll go ahead and get this started. My name is Richard. I graduated from high school way back in 2000. Spent some time in the Army before I went back to college a few years ago, got a degree in biology, and now I work as a fermentation technologist at Corteva Agriscience. With me is my son, James. He's in middle school here in Brownsburg. And joining us today also will be Moira. She graduated from high school in 2010. Wow, makes me feel pretty old. Went to college in Washington, D.C. Chemistry, liked chemistry so much, she got her PhD, she's a doctor of chemistry. After graduation, she also got a job at Corteva and works in formulations, research, and development. Moved here about 1 year ago. So our experiment today, give me a second here. I'm going to share my screen and pull it up. It's called Enzymes and Applesauce. All right. So not everybody knows what an enzyme is, so we'll give a short description here. This one in particular is something called a pectinase. And a pectinase is an enzyme that breaks down a polysaccharide, which is just a word for a big sugar, which is located primarily in the middle portion of cell walls. Pectin compounds are found inside plant tissues, especially in fruits. They're large molecules which are responsible for keeping particles in suspension in fruit juices, kind of like oil floats around in the water in Italian salad dressing. Pectin can influence the juice's thickness. And when most people buy apple juice, they don't want it to have a bunch of cruddy stuff floating around in it, right? They want it to be nice and clear. So when processing apples for apple juice, they'll use pectinase to break down those big molecules, and then they can run it through a filter and it's a nice clear applesauce. We add pectinase to that to make this happen. Today, we're going to kind of mimic what happens in juice processing plant and try to get ourselves some applesauce, or apple juice from some applesauce we bought at the grocery store. If you guys want to do this at home later, just a quick search on Amazon. I was able to find I don't know if you can read that, a pectinase enzyme. It's called pectinic enzyme. Cost a couple of bucks. You just need some regular cheap applesauce from the grocery store. And I know the experiment for, looks like we got somebody else. Awesome. So in the experiment procedure, it calls for graduated cylinders. We're not going to use those today. I was also able to find some graduated plastic cups. I don't know if you can see that. They've got lines for measurement on them. And we're going to be using those in place of solo cups and graduated cylinders today. So we have applesauce. We have one labeled A, one labeled B. We have our pectinic enzyme labeled enzyme A. And for a control, what we use a control for is we want to mimic the experiment in every way except for the thing that we're testing. So today we're testing the pectinic enzyme to see if it actually works. So for our control, we're going to add a tablespoon of salt. So we're still adding a tablespoon of something to our applesauce. And when we're done, we're going to filter our applesauce. We're going to filter our applesauce. Whoa, what happened there? Okay. And see whether the we'll keep admitting. And see whether our pectinase enzyme actually does help us produce more applesauce than our salt, which will be our control. So, son James here will add our pectinase to our applesauce. I'll add the salt. Right. We're going to stir it up real well. Make sure you get it all into solution into the applesauce. There we go. Maybe you can see a little bit better what we're doing here. All right. You got it all in there?
Yep.
All right. So now we are going to set those to the side and set a timer for 10 minutes. And while we're waiting on the enzyme to work, we're going to learn a little bit about cell biology and a little bit more about enzymes. So I hope everybody can see my screen all right here. So for today, we're going to compare our cell to a microbial factory. So we've got roles in a factory, kind of what goes on in a cell that compares to it, and then the functions. So we've got, in a factory, we're going to have support structure, walls and ceilings to hold everything up so everything can stay in the same place. In the cell, we've got a cytoskeleton that's made up of tiny little tubules that kind of go throughout the cell and make sure everything stays where it's supposed to be and help to maintain. And a factory will also have a shipping and receiving department, right? So things that need to come into the factory to make stuff can get in, things you make can go out and get where they need to be. The cell uses a plasma membrane that kind of surrounds the cell and selectively lets molecules and other things through the membrane into the cell, out of the cell, and controls all, just regulates whatever comes and goes. Factories will have a chief executive officer. That's somebody that makes sure everybody's doing what they're supposed to do. In a cell, we have a nucleus that contains genetic information that tells everything in the cell what it's supposed to do. It controls the cell activity, determines what proteins to make and when. It'll be some sort of a power plant in a factory, whether it's just electricity from the city or a steam generator to make things move and work in the factory. And in the cell, we've got mitochondria and chloroplasts. Chloroplasts will be in plant cells. Now, these transform one type of energy to another type of energy that's used by the cell. Typically, this is ATP. And plants will take light energy and transfer that into ATP, and other types of cells will typically break that down from sugars. All right, we've got a factory floor. In a cell, it's cytoplasm. It's like a gooey jelly that fills the entire cell that everything can move around in. And it contains organelles and just about everything the cell needs. And assembly line workers in factory to put things together. And you also have that in cells. They're called ribosomes. And these will take the instructions from the DNA, from the CEO, and will build them into proteins that the cell can use or export to be used by other cells. And these ribosomes, many of them are found in an assembly line, which is called the endoplasmic reticulum. And this is where the proteins are prepared for use in the cell or for export. In the factory, we've got a break room. In a cell, we've got vacuoles. These are just tiny little bubbles inside the cell that can be used for nutrient storage and also for storing and disposing of waste. And of course, we have a maintenance crew. In the cell, these are lysosomes. Maintenance crew will repair items in a factory and break down things and clean them. In a cell, lysosomes will float around, find trash, discarded pieces of whatever, and they'll just kind of digest them, eat them, and either take them to the cell wall and throw them out, or like sometimes if it's reusable, they can release it back in the cytoplasm. All right, so how many cells or microorganisms are present in the human body? It's estimated that an average person has about 30 trillion human cells, you know, hair, skin, organs, cells making up your teeth, whatever, and 40 trillion bacterial cells. So we've got 10 million more microbes living in us than we actually have us. It's kind of interesting to think about. So that means there are about 13, let's see, for every 10 human cells, there are about 13 bacteria or other type of microorganisms present. And researchers believe there are more than 10,000 different species of microbe that live inside of us, making up their own ecosystem. And somewhere between 81 and 99% of these species have been identified. So there's, you know, still a few we don't know what they are or what they do or what their purpose is. So on to our enzymes. So enzymes are substances that are present in every animal cell, plant cell, microbial cell, and act as a biocatalyst by either starting biochemical reactions or making them run faster. They're composed of two parts, a big kind of well, when you zoom in on it, I wish I had a picture of one now, but I didn't think of that beforehand. It just looks like a big knot of protein, just strings of amino acids wound into each other with a non-protein portion, which is either a coenzyme, which is a biological molecule, or a cofactor, which is non-biological, kind of like the vitamins you take in your Flintstones gummies, or if you do take vitamins. Enzyme's job is determined by types and ordering of its amino acid strings, as well as its shape. It doesn't lose activity after it performs their job. It can be used over and over again. It's estimated there are approximately 75,000 types of enzymes that exist in the human body. And each one has one specific job, one specific reaction or molecule that it acts on. Because they have so many applications, scientists have found it helpful to classify them based on what they do, what substances they act on, and the reactions they accelerate. There are 6 key groups: hydrolases, isomerases, ligases, lyases, oxidases, and transferases. Those are big words. I don't expect anybody to remember them. It's just, good to know. Sometimes it's fun to hear big, sciencey words. And they each have a different job. Now, like we've already mentioned, they're specific. They only have one thing they do. Each type of enzyme only reacts with one substance. It's important that enzymes don't cause reactions they're not supposed to. They've got a special pocket on their surface called an active binding site that fits perfectly to the molecule they're supposed to react on. And after the reaction is complete, then the new molecule, takes the molecule, changes it into something else, and releases it out into the cell, and it's transported wherever it needs to go after that. Things that can affect enzyme performance is mostly the environment in the cell, or in this case, in our apple juice, right? It can affect the speed of the transformations they perform. If the conditions are bad enough, it can change the actual shape of the enzyme, preventing it from performing its job. When this occurs, we say that the enzymes are denatured. So things that can affect them are temperature. Higher temperatures may speed up the process until it gets too high, and then the process will stop because the enzyme changes its shape and actually can't fit on the molecule it's supposed to react. pH can change. So if it gets too acidic or too basic, that can also affect the functioning of the enzyme. And the concentration of the substrate, which is the molecule the enzyme is supposed to act on, can influence the reaction time. All right, so when the enzyme's done doing what it's doing, how does it know to stop doing it? One of the most common ways it does this is by something called feedback inhibition, where the molecule the enzyme is actually creating binds the enzyme and shuts it off. They bond with the enzyme at a binding site, causing it to change shape. That way, it just can't do its job anymore. The key purpose of feedback inhibition is to prevent too much product from being made. Molecules called activators can restart the reactions when the enzyme needs to go back to work and get everything started again. All right, so we are back to our experiment. We've still got about, let's see. Okay, there we go. We've still got a couple of minutes left before we can harvest our apple juice. So now would be a great time if anybody has any questions or predictions of what they think is going to happen with our Pectinase enzyme or with our salt control that we're calling Enzyme B. Feel free to put them in the chat. All right, no questions. James, do you have any questions? Oh, we got one. Yes, we are waiting for the enzyme. All right, we got about 25 seconds left, so I'm going to take a chance and go ahead and filter it and see what we got. So for this, I just grab a couple of coffee filters. My assistant James is going to be filtering the Pectinase enzyme, Enzyme A, and I will be filtering the applesauce, our control, with the salt that we're calling Enzyme B. I'm going to stir it up one more time. I set the stir over to the side. Let's make sure we get James in the picture here too. Just going to wrap my coffee filter over the cup. And just pour to see if we get any juice. Hold your cup kind of like this, buddy. There you go. It's going to take a couple of minutes. It's filtering kind of slowly. There you go. If you turn it all the way upside down, the juice kind of almost seals the coffee filter. It doesn't let any air into the cup, so it kind of creates a vacuum and won't let any juice out. All right. So that should be good enough. All right, so we've got our enzyme B, which was the salt, and our enzyme A, which was the pectinase. And as you can see, there's not a lot of juice, but the pectinase enzyme roughly, I would say, doubled the amount of product we got from the applesauce. And that's our experiment. Do we have any questions, comments, or concerns now? Feel free to put them in the chat. Oh, you're very welcome. It's been fun. We're kind of expecting a few more viewers, which was fine. We didn't get them, so we've got a little extra time. Moira, did you have anything you wanted to add?
Well, I guess my question would be, why was the salt so much worse than the enzyme? What about it made it where we had less juice?
Well, the salt really didn't do anything. Actually, it instead of helping the juice to come out, it may have reduced it, because when salt gets into solution, it breaks down into 2 ions, and it takes some water to do that. So I wouldn't say it absorbs it, but it does kind of sequester the water a little bit. And it wasn't doing anything actively to release the juice, where the enzyme was breaking up larger molecules, which was making the juice thinner, more liquidy, and make it easier to filter.
Thank you, that was great.
You're welcome. All right, well, that is the end of our presentation. I hope you guys enjoy. Oh, does salt have a lot of molecules? Like many. Yeah, salt, each little molecule of salt actually is made up of 2 smaller molecules called ions. For table salt, it's sodium and chloride, and one's negative and one's positive, so they like to stick together real tight. When they get into water, water is also very polar. It's got kind of a negative charge to it, so it helps to pull those ions apart and they go into solution. So for every molecule of salt you put in water, it turns into 2 separate molecules. That's a good question. All right. If there are no more questions, y'all have a wonderful day, and look forward to doing this again next year. Enjoy the rest of Celebrate Science.